So good morning, everyone. It looks like we are all coming online. Good to see you. Happy Wednesday. We'll wrap up the discussion we started on clustering last time today, and we will use part of the time to do a lab session. And so appropriately, the lecture is headed wrapping up clustering and plus lab session. What I would like to do today to begin us off is to go back with a warm-up question like we always do. Jessie will have the honor of writing today's question. I don't think she has ever written a warm-up question, so be ready to write it up. I remember on several occasions when we had a topic, I would ask you to tell me what comes to mind when you hear a word. And today's word is clustering. And you can think the way in which we are looking at this, but you can also, your mind may go wherever it may like to go. And I've got pictures that show clustering in a good way. We have groups of things there. And so let me see what you have to say. And I remember when I asked it the same question about explore, when we talked about exploratory data analysis, there was some very fun answers. And Dora was one of the answers. That remains one of my favorites. So let your mind go wherever it may. And clustering is a topic. You hear the word clustering, what comes to mind? Jesse, go ahead, write up that question and you will type it up one more time later on. Another thing I want to tell you is at the end of every lecture from starting today and what we will have on Friday and after Thanksgiving break, we will have what I would call an in-class exercise. Just a simple question that you can participate in at the end. We will use the chat feature because we don't have assignments. That's a good way to think. And it will be relevant to the topic that we have discussed. No worries, no stress, just focus, follow along, and you should be able to answer whatever question I may have. So be ready for having a question like that. So I will save a minute at the end to do something like that. And so I very strongly want to encourage you to be attending classes. It could count to your participation, but if you don't get a chance to show up for whatever reason, let me know ahead of time. You don't have to worry too much, but I don't want you to be relying on just watching the lectures afterwards. And so it would be good to get that. Let me go get started. Before I do, let me see if there is any general question or anything like that anybody has. You could use the chat feature, but you can also unmute and ask me. Does anybody have anything very general before we get started that you would like to ask? So I remember there were questions around hierarchical clustering last time, two of them, in fact, which will be the starting point for what we will discuss today. Those are taken care of. Those of you who have asked me, relax, we'll answer them. But do you have any questions around the class or whatever else before I get started? I'm also watching, sometimes I turn my face. This is where I see the gallery of those who are attending, and so I may turn my face sometimes. Any questions before I get started? Okay, we're all good. Okay, let's go then. So just to be clear, what we have done and what we have reduced the discussion around clustering when it comes to methods is to two of them. There is a very large number of clustering algorithms around, and I would like you to have at least this two methods mastered and also see where they are successful and where they fail, and also try to see how other methods may come into the picture. So what we did last time, other than discussing clustering in a general way, was look at most obvious or one of the go-to methods or one of the most intuitive methods for doing, and that's called k-means clustering. And the means there is average, centroid at the end of the day. And so the k is how many clusters you have. And one of the features of that algorithm is you need to know ahead of time how many clusters you have, which is sometimes a limitation because we don't know how many groups we have. In fact, that is one of the things we would want to discover. And so that algorithm has got that disadvantage. But if you, in collaboration with other methods or domain knowledge or something like that, have an idea for k, which is partly something that this method can give us a clue, then you could use that k to find out what is the best clustering for that particular k. And so that was the idea. It is intuitive and it's simple, and it is successful in the sense that it does two functions are simultaneously optimized. The centroid is completely all the time moved around, and an object is placed to the closest cluster that we have assigned it, and we begin things randomly. Hierarchical clustering, on the other hand, does not require us, and we started discussing this, and this is where we stopped. And so the idea here, both in k-means clustering and hierarchical clustering, for that matter, for any clustering, you would need to have an idea for having to measure similarity or distance. In fact, that is at the core of it, right? So you're grouping things that kind of are similar to one another into one group. And so the case is true for hierarchical clustering also. This time, we view the word, and the picture that was on the cover of the slide on the left was showing you hierarchy, and that's what I was trying to get to, is you view the word in a hierarchical fashion. That means the clustering that you come up with are nested. So you group these things together. If I am allowed, and I'm going to relax it a bit more, then this can't belong to this other one. This one can't belong to this other one. There are times when such an approach, such a view is the right thing, and there are times when it's not. I'll give you an example, especially when you get to the lab session, but let's assume that that is the word. That's what hierarchical clustering would do. And so we would start by saying everybody is on their own. So each item we have is a cluster on its own, and then we start to group. And this is confusion. And so the ones that are the closest, depending on whatever measure we have used, will be fused together. In this case, A and C, we will start the story. We have gone through this one last time, are the closest to each other. Euclidean distance is, as I said, one of the most common ones to do, but you've got an opportunity here, depending on what you're trying to do, to define your own distance measure. And so if A and C are the closest now, at least in Euclidean space, they get joined first. And then once they are joined, we have a cluster, and then we see D and E are close to each other. That will be our next thing to identify. And you can see now in the dendrogram, the height at which D and E are joined is slightly higher than AC. This is a direct measure of the distance. So now we have an information captured in this dendrogram that should be interpreted closeness along the vertical axis, which is this what these numbers are showing you. So you've got the blue, and then you've got the red. By then we now have opened up an opportunity to measure distance between an item on its own, like A and C were and D and E were, then potentially another item and a cluster. And that opens up a question, which I got asked last time, which we will discuss today. How do you measure if I have an item and then a cluster? So generally speaking, the distance that we really are measuring is a distance between clusters. And that's non-trivial because you've got many elements in a cluster. What does it mean to say this? How far apart are they? Even when I have settled for a distance measure, like Euclidean distance, when do I say, how do I calculate it? We'll get to that. And so you will see now the next time between A and C as one cluster, and B, that's where the next fusion happened. And then at the very end, the whole thing comes. So hierarchical clustering will start with N, all the items you have in the leaf, and it will end up with one cluster where everybody is put in one big thing. And this hierarchy is now what the dendrogram represents. This is where we stopped. And we also looked at this fictitious example with 45 observations. And as we usually do to make life easier for us to visualize, we think the world is two spaces. So we've got only two features, x1 and x2. And here is how the 45 observations are in a scatter plot. And the color shows you what groups they are. And we've used this. And so the dendrogram now, after it is built, would be up to us to decide or how to drive a clustering from it. And the way to do it is to decide where to cut the tree. And so if we cut the tree at the very top, like, everything being together, then we have got one clustering. That's not so very meaningful. I mean, we just say everything is one cluster. I ask you to cluster this classroom, and then you tell me the entire class is a cluster. You could do that, of course, but that doesn't help. But the visual of this is already helpful. So we can see how they are. We just decided to have it in one cluster. If we cut it after we look at the tree, in fact, some of these things, you may be surprised, we do them, or people do them visually. So they get a dendrogram like this, look at it, and then decide where to cut it. There is some automatic tools that would help decide this. You can get some exploration that way, but the visual one is not unusual to do. You just look at it, and this looks like it makes sense to kind of have to, but then you can see maybe the right arm of this one has got the majority of the class, and the other one is not, kind of smallish, and you may think, no, that probably doesn't fit what I want, and so you do cut it at a different level. Now you get three, and these are actually the three observation or the tree, the fictitious example I showed you earlier would map to this. And hierarchical clustering does a very good job on this particular case, identifying which ones they are. It's one thing to decide the height, and it's completely another thing to decide which cluster each one of these items would belong. And hierarchical clustering there does a good job. Now, this is just an explanation. That's why I flipped through. This is just a word version of what I explained in the figure. Here is one example that we didn't talk about this one last time. I want to tell you about, and this has to do with avoiding to do misinterpretation of what a dendrogram is. Here is a data set that has got only 9 elements in it, and there are only 2 features, X1 and X2. And what you see on the left is a dendrogram. And if you were to look at the dendrogram, which sometimes is just as useful in terms of summarizing your data as it is for doing classification or grouping later on, you should be able to understand it the correct way. Sometimes it gets misunderstood. So if I see this dendrogram now, somebody may wrongly feel like item 9 and item 2 are kind of close because they appear kind of close to one another in the leaves of this tree. That is very, very wrong. You shouldn't have anything to draw from how things are laid out horizontally on a dendrogram, because that is just a matter of choice, really. If you have a tree, let's say 90% of this class or 80% of this class either computer science students or computer science literate. So let's say I have a tree and I tell you there are leaves, and I tell you there is n of them. How many different ways can you draw the same tree? It's essentially the same information, but you've got different ways to show me the leaves. They are the leaves, and you can show me how they are, who's the parent of which one is, and so on. But it's not only one way I can draw that tree. I can draw it in a number of different ways by having to choose how the leaves are ordered. And if I have n of them, in fact, I have exponentially many, to be quite frank, many ways in which I can order. Exactly the same tree, the way it is visualized when I look at it is different, because I called 1, 2, instead of 1, 2, I call them 2, 1. They're children, the parents the same. All I have said is the children are 1 and 2, but I could write 1 first, 2 next, or I could write 2 first and 1. Think about another place where there are 3 children, 1, 2, 3. I could have this in any order, 1, 2, 3, 3, 2, 1. You get what I mean, 1, 3, 2, and so on, right? So for that reason, there is actually very many ways in which you could have drawn the same dendrogram. And so the horizontal axis, or the way in which the leaves are ordered, that should not make you draw any conclusion. 9 and 2 are very far apart. The only reason they have come close now is because of the way we displayed them. What you should look in the dendrogram to see similarity rather should be on the vertical axis, which is what the height would tell you. So 1 and 6 are the first to be joined. You can see the height is short, and you can see now on the display on the right, 1 and 6 in fact were close, and so are 5 and 7. You can see that they probably they're about the same distance. 1 and 6 and 5 and 7, they're about the same, you can see, and they are so. Look at what 9 and 2, they're so far apart, really. The only reason they look closer here is the way we draw them. What is close is 9 is close to whatever it is that we grouped as 2, 8, 5, and 7, to that entire cluster, to that entire subgroup. To that group, yes, 9 is the closest compared to other things, and that's why it's fused together with it. And so that's the thing that I wanted to tell you before I leave the topic of hierarchical clustering. And this is another elaboration on what I just told you and how they are. So remember, the vertical axis on the dendrogram is what tells you about similarity, not the way in which things are displayed. This is the place where I'm going to go tell you now the second question. Let me actually answer the first question I got last time about the relationship between dendrograms and phylogenetic trees. Okay, for those who do not speak biology, phylogenetic trees are trees that kind of show you how evolutionary things have happened. They are very, very close to dendrograms. This is where you can measure, similarity between objects to be like mutations or some changes in the biological sequences. And if you think about two things being very close to one another, you can imagine that you can fuse them to find a parent. That's where they came from. And so you could put that one in, and then you can build up this hierarchy. And the hierarchy becomes very meaningful now, and they are used. And the example we will use today in this lecture, also in the lab session, would actually be on genomics. And so there is a close tie between what dendrograms are and what phylogenetic trees are. What's the difference? Dendrograms are very general. What your distance measure could be anything. We are using now to talk about similarity between students in a classroom or people shopping in a mall or whatever else as there are. Whereas in phylogenetic trees, it has inherently about evolution, about changes that have happened over time. When you have specialized it to that and distance is measured in terms of changes in sequences or other hereditary information, then they are equivalent. That was the answer to the first question. Now comes the second question. Okay, we have convinced ourselves, and if we think a little bit more mathematically now, the finding of distance between objects when we build hierarchical clustering could be said a distance between two clusters. In the very beginning, a cluster is just an item, and so we could talk about similarity between items, which we have discussed so very many times. And so we could use one of very many choices, and let's say distance is measured in Euclidean distance. That is between objects. What happens when we have more? That means now, whether it's 1 or 2 or 3, we can always talk about how do we measure distance between two clusters. And we have a bunch of them, or 4 of them that we'll discuss here. And this depends on how we interpret distance to mean. And by that, I don't mean to interpret in a way like, subjective. I like it this way, somebody else likes it that way. I don't mean that. I mean what we choose to measure as a distance between clusters is we have options, and depending on the application, one thing may be better than another, and the dendrogram we would get would also differ. And so it is a significant decision on our part to make, and this was the question I got last time where I stuck. And so let me go through each one of these. So we've got lengthy or wordy descriptions, and I'll come back to them, and I think a picture will be the best way to talk about this one. So I'm going to go to the next slide and then come back to this one to explain it or to tell you the connection. So what we are doing now is trying to decide what is the distance between cluster A and cluster B. Cluster A, in my toy example, it's got 3 elements, and cluster B has got 2 elements. And these elements, I have shown them in blue. So for the moment, forget about the red ones. And so we've got 3 things here and 2 things there. What is the distance? One view may be to say, I am going to call the distance between the clusters as the worst distance possible. What are the furthest points between A and B? And that is what you see in the bluish kind of thing. It's called complete. So that's the worst case. So I've got these elements, and I compute these distances and find the one that's the farthest apart. That's how far apart they are. And it kind of would give you a sense. This is your worst case. And we do this most of the time in all kinds of analysis in computer science, including runtime. We care about the thing that even though it appears only very rarely, that will be our worst case, and we will be safe. That will be an upper bound. This is the way we can think of about the clusters. So worst case, complete distance, the maximum distance possible between any pair. You could take the opposite and then say, I would care for the one that kind of brings me closest, and so look for the one that is the closest among all the possible pairs of distances. I will call the distance between A and B is the minimum, and that will be the other extreme. And there may be times when you will settle for this. That will be a better choice for you, because you could argue that this cluster comes this much close. If only we had one representative at least that has brought us. Let's call that one the distance, and that would be called single. So we've got 2, complete and single. These things are linkage. Don't ask me about names. Sometimes names are historical. This is what they call them. So let's call them that. So linkage is what these things are called. And the first one, that's about the worst, the maximum distance, is called complete. And the other one is called single. I really don't know why they're called this way, but they're called this way. Then we could also think about an average. I could, this would be a happy medium, right? So you have the worst, you've got the shortest, but I could also compute all these pairs, these distances, and then divide them by how many pairs I have, and that will be the average. And it's not easy to show on this picture I have, so I left it alone, but I just told you averages, you add them up and divide by how many you have. The n, by n here, I mean the number of pairs of distances you have. So that will be average. The other thing you could do is instead of average, you have a cluster, I've got another cluster, I've got the elements, but I could compute the centroids, just like I did in k-means. And I say, now here is the center of gravity for this cluster. Here is the center of gravity for this other cluster, and I have shown them in red. Now I take the distance between these. That's called the centroid. Okay, so now here you have the word versions of complete, single, average, and centroid, and these are the 4 ways in which you do. One thing, the answer to the question I got last time is we have choices. These are the 4, and the most common ones are complete and single, I think. But depending on the application, you may choose to do some other things. This is how they are computed. So they are, in fact, computed. All of these pairs are computed. Even to find the maximum, you need to compute all of them. For the average, of course. For the centroid, you have even more things to do. But you don't have, as a user of a certain tool, to be doing this manually yourself. So you need to specify them as a parameter is what you would do. Otherwise, you don't have to do this calculation yourself. Of course you can, but they're usually available. Okay. That was what I wanted to talk about hierarchical clustering, and I need to probably tell you now something extremely important. The distance measure you would use when you have items would be application specific, and you will be sometimes, that's probably where your innovation may come in. What did you use? If you remember the K nearest neighbor classification, I told you about the restaurant and the tips, and you are classifying them. It was not so obvious how to say, how to group things together. The K nearest neighbor algorithm is very close to accept that there is classification. So the big difference, as you all know, between classification and clustering would be classification is you have supervision, you have labels, here you don't. And that's the biggest difference. But colloquially, people, when they talk about this, they also say classification when they actually mean clustering, which is fine. But the idea there was that you were trying to group things that are, similar by their distance. And you remember that tips, you turned it into a vector, and you saw similarity or agreement and disagreement, and that was your distance measure. So there is a number of different ways you can come up with to define distance. But let's say you have settled for that and you think the whole world problem is now solved. No, sometimes you actually would want not the distances themselves, but the profiles. And a good way to give you an illustration for this one would be to take you to shopping. So you've got a place where people would shop, and see this a place where you are asked to come up with understanding your customer base, and the company has hired you, and you get some data about these people and their purchasing behaviors. So you may see that there's people here, and maybe let's say how much they bought, how many things they bought, and how much they bought is what is recorded. You may now then see one person has bought a certain number of things. In one case, let's say they were buying socks, or this is what the book is, by the way. I probably should have told you, we are using the book's examples and things like that. So if you want to further read, chapter 10 of the ISLR book is where these things would be. So there is a little bit of a discussion around this if you wanted. So let's say you're looking at behaviors between two customers, and you see how many socks they bought, how many TVs they bought, and that example will show up later on, and how many other things that they have bought. If you were to now see the distance between customers, because you want to group them, you could either talk about how much different they are in each of these items that they have bought, or you could see their profile, how many things that they have bought together in a way. So you've got, like, for example, the green one is one customer, the golden one is another one, and the purple one is the third one. If you were to go just by distance, you may feel like the golden and the purplish are close, because if I take them pair by pair like this, they kind of are close. The Euclidean distance between them is close, and this might give me the wrong impression. But take a look at what is happening with the green one and with the yellow one. They kind of have very similar patterns. They buy some things together in a way, and then they have except for the magnitude, they pick on the same kind of pattern, and they dip down on the same kind of pattern when they are plateau, the same kind of pattern. So if you were to segment these ones, what will be more important for you is to see the correlation between them rather than the actual distance. And so now your algorithm may group green and yellow together, whereas the purple one, or whatever this other color is far apart. But if I was to go just by Euclidean distance, I would have gotten a completely wrong information and then would have grouped them together. So in some cases, this will be typical of time series, which is the subject for our discussion on Friday. Those of you who are working to time series data mining is a relatively big topic. We'll have one nice, good session on Friday. We'll talk about that. This is bread and butter there. So when you have time series, you can in fact think of this as time series, as things that have happened, measurements that have been taken on different time points in a 24-hour period, for example. And it becomes so obvious then, if you had 2 or 3 or 4 series to put together, you would not go for Euclidean distance. You would go for some kind of correlation between them and some way to normalize them and things like that. Okay, so that's one of the things that I wanted to talk about. The other thing I want to talk about, and we'll get back to the same purchasers in that store who are buying socks and computers, is to scale. Sometimes when you do clustering, your results will be heavily influenced by the scales of the things that you are doing. So you do 2 things. One is you normalize. You normalize by or you normalize it so that the mean will be 0 and the standard deviation will be 1. So if you do that pre-processing, then you get the essence of it and you will not be influenced too much by the variations within themselves, right? So now you will be able to compare one against another. That's one. The other thing is about the scale itself. People who purchased socks and the people who purchased computers will tell you very different stories. In this particular example, there are 8 customers. Each one of them has got its own bar. And what you see on the left is how many they bought. And so these 8 customers really differed so much on how many socks they bought in this Walmart shop. But they're kind of similar. Some of them didn't buy any computer for that year, and 5 of them did. But it was cool year, and we don't expect them to come back and buy another computer that same year. That's why everybody was 1. And so what you see here now, if you did not normalize the picture you would get, and if you use this one as a basis for your distance measure in your hierarchical clustering, you would be forced to draw a very different kind of conclusion. If you were to scale them though, then you would get the picture that you see in the middle, which is a much better telling of the story, because now the significance of the computer purchase has gone up because you have normalized into the mean and the standard deviation taken care of. So the middle one is what you want to do. What you see on the right is not what you want to do either. And that is when you look at the raw value for how much money they spent, and the people who purchased socks disappear because these other guys are paying computers, and so you can see the difference in magnitude. And so if you did go with the normal, with the, what is it called, the magnitude of that thing, the actual dollar value for it, then you get another kind of information. But this is about, customers and so on. So you don't want to necessarily be looking at the ones that are buying just expensive things. And so what you want to do is have a picture of the middle one. And you do this when you do clustering all the time. And it is a good practice to do. Before I get there and tell you this, I should caution you. The way I presented this one now sounds like scaling is the right thing to do all the time. That's not true. There are some, in genetics, for example, which we'll see an example of today, where you would probably be advised not to do because of the minor, details that you are interested in, the gene expressions and things like that. But if this thing has, if you have enough information about it, and if you do not have any compelling reason not to use it, scaling is the right thing to do. That's probably a fair statement for me to make. Okay, so I think we have, discussed quite a number of different things. I want to maybe kind of remind what practical issues we need to do when we do clustering. The standardization is what I just talked about. That's one issue, one thing that you need to decide whether you need to standardize your variables in some way. When it is hierarchical clustering, whether the dissimilarity measure what dissimilarity measure or distance measure should be used, and what linkage among the 4 ones I told you. Complete and single are the common ones, and we will see an example where visual aid would be a good way to get about it, but that's a decision, a practical issue to answer. And how many clusters do you choose when you use k-means? that is a difficult question to answer. We don't have an agreed upon solution. And I can point you later on to methods that you could use that are more advanced. Okay, let me use a little bit of the time now I have to give you a concrete example. The discussion in the book is, I think, a good one, but there is also a reference for those of you who really wanted to read of a paper that appeared many years ago, but it is significant. It's a big deal, and the method that it introduced is even more important than the biology or the cancer study discovery that it had made at that time. So let me take a bit of a moment to tell you about this, and I will, then go to a lab session. This is gene expression microarray study. This was a study made at Stanford, and the publication appeared here. And then, as I said, more than the study about breast cancer and what it found, the method it introduced is significant. And so here is there are 8,000 genes, and there are patients, 88 of them, that are involved in this study. And after they have done what they have to do, medical knowledge and biology and whatever it is that it needs to dictate, two decisions were made for this hierarchical clustering approach. Hierarchical clustering lies at the heart of this study. And so the two decisions, like the practical issues I talked about in the previous slide, were to decide on what metric to use for distance. And they decided to use correlation, not actual distances, fine. And the other thing is the linkage, the distance between clusters when the hierarchical clustering is done was average, not complete, not single, but average was the one that was used. One biology fact you should know here to understand this work is that there's 8,000 genes to worry with. And so this distance is to think about. And so there's only 88 patients. That's not very many, but the genes are so much, 8,000. Among these, they had to do the ones that would be important to look at. This might be something that you would want to know. This is biology, and so you may have one way to get to important features, but this is a common data science problem. I've got a problem, data set, it has got many features. What are the ones I should look at? And we do this in many ways. And I told you, ironically, one of the things that clustering you would want to do is to get handle and to have got smaller groups so that you could just further things with them. But you wanted also to use important things to do your clustering. And so if you are talking about 8,000 and that order of magnitude for features, you better find a way for you to help you narrow this down. And these people identified 500 of them. And these 500 genes were chosen because they were intrinsic. And so this is breast cancer, so it's about women. And so they were trying to look for genes that would vary the most within, among different people, different women, but they are kind of intrinsic to that woman because their variation within that person were not much. And so they had a way to get down from this 8,000 to find 500 intrinsic genes. Okay, so we've got population size now of 88. And we've got 5 things to worry about, and we're going to do hierarchical clustering. So let me show you. And this is this was my punchline. I told you that the method was significant for what it did, or the study for what it did in terms of method okay. They're equally important. Whatever they discovered about breast cancer was important, whatever they found in 2003. But this is a summary, you can call it, of a data set. And so what are we seeing here? First of all, you could look at this hierarchy showing up. So there's some dendrograms. The 88 women I told you are now in the matrix called A, and they are the columns. And the rows are these 5,000 or 500 genes. And you see these colors, green and red. This is wherever something is expressed, as they call it. So is this like more than average? Are you seeing a lot of it, or are you seeing not enough of it? When it is high, when it is expressed a lot, it gets the color red. When it's not expressed that much, when it is low, it gets the color green. So red is bad. So if this was, people you're looking at, whenever you see red, that means that person has got that cancer. Okay, when it is green, they are negative, is what you see. Okay, so the 88 women in this study are done, there is a clustering done on them. So there is a dendrogram that you can see hierarchical clustering applied on the women. So the columns of this matrix are clustered. And then the rows, the genes, you could also look at the genes and talk about similarity and things like that. That's also clustered. And so the rows are also clustered. So now, after the clustering is done, this matrix that's 500 by 88 is reordered so that you've got these green batches coming close to one another and the red batches coming close to one another. If you didn't do that, it would have looked at a checkerboard, and so that the greens and the reds would have been everywhere. So after you do all of that, now you get this picture. So before we do anything else, you could say now that I have an excellent way to just look at data. There was a 500 by 88 things that what I was given, and so there are measurements, and there was, these things I've done. I have information now that you can see is summarized. I'm sure very many of you have got something similar, either in your project or in your research. Something like this might be worthwhile. So this is a dendrogram and a histogram at the same time, because this green and red is not just green and red. If it is above a certain value, you make it a green, but it is not always the same green. It's not always the same rate. So you could, shade that. That's what a heat map would be. And so they are grouped together. And so the ones that have expressed themselves have come like this. So what you see now on the dendrogram, that up at the top is about 8 or so, and they have given them names. And this is, what the domain knowledge would tell them. So there is normal, brush-like, and there is basal subtype, and the EBRB. Don't ask me what they are. So there are certain things. And so there is some grayish thing, but there is a certain group that is like where more of these things are appearing, and there is a certain group where it is not appearing. So that was the discovery. That's what they found out. And so they made that relationship. And there is a little bit of an interesting study here for, among these groups that I have shown you, which ones have got a better chance of surviving. This is a terrible disease, and some of them, unfortunately, would not survive it. And so there is a relationship that they found. How many of these actually survived and how many of them were not surviving? That's what this picture is showing you. Okay, so I hope you have understood what I said. If you haven't, you probably would want to go back and read a little bit. But the hierarchical clustering together with the dendrogram together with a heat map was an excellent summary, is what I wanted to say. All right. This brings me now to finishing what I wanted to talk about, unsupervised learning. And that was primarily about PCA, which we did before the midterm, and clustering, which we did in the last 2 lectures on k-means clustering and hierarchical clustering. There are very many other types of clustering methods: solve organizing maps, independent components analysis, spectral clustering, which is a good topic to know something about, are all methods. And I can point, I don't think for this class there will be time for us to discuss them, but know about them. And if you need to use them, you should probably spend some time to learn about use. Okay, that brings me to the conclusion of this part. I'm going to stop sharing here and start doing a little bit of a lab session. We've got some time, but let me see if you have any questions. Okay, let me do the lab session in a way that probably would not be as much as we used to do, but let me give it a try. Here we go. Okay. So Brit, can you see my screen? Perfect. I asked Brit because her camera is on. So what I have here is a very simple illustration for how to use k-means clustering and hierarchical clustering. At the end, I have a piece of code that's taken from the book that talks about the breast cancer type of data, but for a much smaller one. It's called NCI 160. I would like you to have it so that you can see how it is done. Okay, it kind of mimics what I just told you. So for the k-means clustering part, we'll run. So and along the way, maybe you would learn 1 or 2 different, kind of R functions to call. We'll make up some fictitious data now, and the data that we will do is normal distribution, a Gaussian distribution. So if you wanted to do something like that, you could do an R norm, and then you give it how many things you would have, and so you will have a Gaussian distribution. And metrics is a function that you could use to call to generate a matrix. So that's what our data set would look like now. Let me see. What do I do to get here? Yes, I've got generated my matrix. It has got 2 columns and it's got 100 elements. That would mean it will be, if it's 2 columns, it will be 50 rows, and it's a normal distribution. So I'm going to create a data set that is distributed so that its mean, the 2 groups will be, their means are separated. So I have the first 25 elements would have this value, and it would add 3 to them, and the second one will have -4. Basically, I have generated a data set with normal distribution. They are separated, so then when I ask for clusters, I expect to get 2. Basically, that's what I have done. This is a nice way to do. So k-means clustering you can do by calling the k-means function. That's what this one shows you. And you may remember I told you can start from any arbitrary number of how things are assigned, but you may not get the same result. Sometimes you get unlucky, and therefore you may get a bad result. For that reason, you want to have an n start, which is a parameter that's greater than 1. If you just give it 1, it will get 1 result only. So we'll do this for 20. Okay, sorry. We did that, and so we have now a k-means clustering done, and we type it out, and we can see that this thing is doing a good job. The first one, the first 25, it looks like are called cluster 2, and the other 25 are called cluster 1, but it just did a perfect job on getting the 2 clusters that we just artificially created. And here is, a plot of them. Let's say we knew this. The way we created it. It was a cluster, a data set that has 2 obvious clusters because the means were intentionally separated so very much apart from each other, and the algorithm discovered it. What would it do? We know that it is 2, but what would it do if we, k-means algorithm that is, if we say we want 3, k was 3, and let's see what it does. When k was 3, we did that, and we kind of find some information around it. I was hoping that the plot would have, okay, I didn't make it plot. Oh, here it is. So we got and start 20, and so it did not a bad job. So for this data set, it decided to split the other one into 2, and so the one that was red now is red and blue. So k-means algorithm did something reasonable. We already know that it was a group that had 2 clusters in it, but we asked it to divide it into 3, and so it decided to take the second part and split it into 2. One thing to just want to make sure that I illustrate here is you may want to see the difference. What happens when you do not have an n start that is greater than 1? That means you have one random start and you are not allowing the algorithm to try out different things and report the minimum within cluster variation. If you remember the discussion last time, that's our measure of how good the clustering is. So let's see what happens for this cluster that we just did when n start is 1 and when n start is 20. When it is 1, the within cluster variation is something that you can probe and find. It's 104. And when we did it with 20, it became down to 97. That is a reduction for sure. And now you are convinced that it helped. And so as a general rule, do not do n start because it costs you nothing. Of course, if you make it like 1,000, it will be bad because you're trying so very many things unhelpfully, and it will take your time. But 20 will be a good number to use for most type of clustering algorithms, k-means clustering algorithms you would do. Okay, I'm going to use 2 or 3 minutes to tell you about how hierarchical clustering would work, and this is actually a good one because now we can see this linkage, the one that's called complete, the one that's called single, and that's what's called average. What are we going to do the clustering on the very same data set that we've used? So we know exactly how we created it. It has got 2 clusters. What would it do for these ones? And the function call you do in R for doing hierarchical clustering is called hclust. And what do? You give it the distance measure, and you give it the linkage. And the linkage is called method. The parameter, you would call it method. And the names are like this. And in fact, that is probably why this thing has stuck with these names, because the people who wrote these functions first have called them this way. Okay, so we've got this. Now we are going to do an hclust when the linkage is complete. We have 1. When we have average, we have another one. And we have single, we have a third one. So we want to see, I want to see how they look like. And so we're going to have a display where there are 3 rows, 3 columns in a row, and that's what this one shows. And let's plot them. Here we have when it is complete. Here we have when it is average. And here we have when it is single. Now you can see for this data set what it is doing, and you can see which one you might have chosen for this particular data sets. The one that is complete looks like it gives, it's terrible to see here because we don't have enough space to display it. That's because my window is small, but you can drag this around and make them better if you like. But let's see what happens when I have the complete one and I cut it at 2. I got this, which is not bad. When it was complete and I cut it at height, I decide to cut to have 2 groups, which I know, 2 clusters, which I know is the right thing to do for my data set. Complete did a good job, and you can see the partitioning or the clustering this algorithm produced. Cut3 is the name of the function. And we could do this thing for this other one, for the average, and that one is not too bad either, but it is making a little bit more mistake, as you can see. So complete sounded like doing the right thing for us, whereas average didn't. And the single one does a horrible job on getting us this, because it was doing the obviously the wrong way we want to do it. When we are looking at these things that are of minimum distance, it just distorts the whole idea of how this works. So for this particular data set, complete seems to be doing the right thing, and you visualize them. What would happen if we decide to cut it into 4, and we can take the last one for having to see this? And it does give us 4 clusters, as it should, and I don't know how much of good feeling we would have about this. We could also see how scaling would affect this particular data set, and we could get scaling by calling this function called scale. And xc here is the name of the variable we have used for having to show that. And here is what the scaling would have to mean. The NCI data set is what I have here, and the NCI data set has a class or a label, if you read about this one, on this breast cancer type, or whatever that particular data set has got, the cancer type. And so you could do the clustering, but then now you have ground truth. So you could see how much of a good job you have done. And I don't want to demonstrate that one here. I just left it for you to look at if you're interested. There are times when something like this could also happen. We are interested in clustering for the purpose of seeing other things, but we have at least a sample on which we could have a ground truth, in which there is a grouping, and we could actually assess how well our method has done. I'm not sure if I have mentioned this one very clearly before. One of the troubles you would have when you have a clustering method is you actually don't know how well you have done. If you know the domain knowledge and so on, and you say here is a cluster, then you have a biological way, a social way, an experimental way to verify it. That's great, but usually you don't know what's right, and so you don't have a ground truth, as they call it. In this particular case, there is for this data set to tell you how well the methods have done. So the end of the story, when you run this one on your own, is that k-means clustering and hierarchical clustering, both of them do a good job on this particular data set. And there is a little bit of an illustration for how you could use principal components analysis also. So the code is there, and my goal was to at least make you aware of it and tell you a little bit about what is there. The PCA calls you see here is even less than what we did cover in class when we talked about the USA arrest and things like that data. So it's not entirely new, but it is nice to see them together, both clustering and PCA done for a data set that you have a little bit more information about. I've got my 2 minutes. Let me see if you have a question, and then otherwise I'll pose my question, which is what we will be doing from now on. Did you follow me along fine? Okay, I'll take it as a yes. And I've got a yeses. Okay, here's my question. And I think a good way to do this would be to answer the question not in a public way, but send it privately to me. And we'll call this exercises so far. Okay, so we have the warm-up question that's public, and these are the question that you would answer at the end. So take a minute and half a minute or something to think about it, and then tell me your takeaway. What is your takeaway from these lectures? And so you could write 3. And you could have a couple words or a sentence or whatever it is. But what is your takeaway? What is your summary now that we've finished this lecture? I'll make it easier for you to make it focus on today's. If you are inclined, you could take the 2 clustering topics together, what we talked about last time and what we talked about today, last lecture and today's lecture. If you are not passed in the last lecture, you just take it today. So give me a 3-point summary of what we talked about today. That's the question. Is this clear? I hope it is. Okay. Take half a minute or something like that and write me. And I'll let you know when time is up. So we have a full minute, 60 seconds for you to answer. Then I'm getting answers. Nice. I'm getting very nice answers. Okay. The reason I wanted it to be private is because I don't want you to think about what others are thinking. Just tell me what your takeaways for you. That's the only reason. So we'll give you another 30 seconds and then we'll officially call it a day and go. So some awesome answers. So we'll do it this way. I'm going to give those people who are typing another 30 seconds or 1 minute they need. For those of you who are done, you may leave. I will see you on Friday. And the topic for Friday is time series data mining.